Okay, so let's start begin. Welcome everyone to the first webinar of CONcISE. We are very happy to have you here in this session. And today we are going to present some interesting talks about some photonics devices in bio applications. I'm John, and with my colleagues Jargis and Chiva are going to host all the presentations here and helping everyone with any issue that we can have. So first at all, before we start the webinar, I want to talk about some housekeeping rules. So the first one is this session is going to be recorded for and posted in our media platform of CONcISE. And if you have any request or concern, you can contact us in the email we put in the chat. Another rule is this session is going to be long, about maybe 1.5 hours. And it's going to be very, very exciting. And finally, we have today 5 different presentations. And after each presentation, we are going to have a question and answer session. And for this, if you have any question, please use the question and answer feature here on Teams, and we can administrate all the question and make everything very easy for everyone. So let's begin. So here you have our pictures of the moderators, Georgis and Chiva. And this CONcISE project is a new European Union funded Marie Curie Action Doctoral Network set out to bring more data efficient and quality oriented techniques for biomedical optical imaging to the market. We are totally 11 doctoral candidates, including myself, Jargis and Chiva, from different 8 European countries with a few funding to take this research approach. And it starts at the 1st of February of 2023 with all the recruitment process, and it's going to end at 2027. The objective of this consortium or network or doctoral network is to develop a novel and conventional multidimensional optical imaging techniques for the mapping of absorption.
Sí, sí, sí. A ver, una vez entres en la rueda ya está. Así que a ver si hay suerte y ya está. Sí.
Sorry, for mapping of absorption, scattering, and fluorescence in biological tissues with visible and IR light through an innovative paradigm based on joint hardware and software development and integration, data compression at measured state without information loss, and finally, adaptive data-driven strategies, including deep learning approach to overcome the actual system limitations, which are typically developed trying to maximize the number of measuring points, for example, pixels in a camera, position in a raster scan, etc., regardless of the information content. So to get this objectives, we have 3 different work packages: the Smart Dot, the Smart Fluo, and the Smart 2PM. The first one consists in a multispectral diffuse optical tomography, and the objective consists in obtaining at the end a 3D map of absorption and scattering on thick tissues. The second work pack, the Smart Fluo, consists in a clinically compatible endoscopy imaging, and we expect to obtain real-time quantitative map of fluorescence in different tissues. And finally, the Smart 2PM, that actually I belong there, is to develop a new high-resolution nonlinear microscopy, and we expect to obtain the prototype of a wide field to photon microscopy. So in this all project, we have some 8 beneficiaries, all the logos you can see there, and 4 associate partners from all different countries of Europe. So thank you so much. And just we are going to start with our first speaker. He is the Dr. Salvador Torres from Fila Laser, and he's gonna talk today about supercontinuous source characteristics. He's a doctor in physics from the University of Valencia, and he also worked first as laser technician in Proton Laser Company before arrives to Fila Laser. His interest in research are the mode-lock lasers oscillators in fiber optics, photonic crystal fibers, and supercontinuous source. Thank you so much, Salva, for being here, and I let you speak to start.
Okay. Hello, everybody. Let me share my screen. Can you see it?
Perfectly.
Okay. Well, as John said, my name is Salvador Torres, and I'm a member of the R&D department at Fila Laser, and I have been here in this company almost the last 10 years. Okay. My role within the group mainly involves the development of supercontinuum lasers and other types of lasers, as well as the design of systems or setups for possible applications of our lasers. So in the following presentation, my idea is, as the title of this first slide says, to show you briefly what a supercontinuum source is and some of its characteristics. I've structured this session in the following part. First, I will describe or define what a supercontinuum source is. Then I will show you a general scheme of the parts of a supercontinuum source, and I will give you a short description of the properties of each one. After that, I will explain the specific characteristics and some applications of two sources, both designed here at Fila. The first one is the ice blink, and the second one is the cycle. To begin, and as a definition, we call a supercontinuum light source a light source that has an extremely broad spectrum, which can range from ultraviolet to infrared. And the other characteristic that a supercontinuum has is that the generation of this broad spectrum is achieved through nonlinear interactions of light with a propagation medium. Otherwise, the halogen lamps would be considered also a supercontinuum source, but is not the case. Since the principle of generation are the nonlinear effects, high power density is required to generate it. And the best way to achieve these conditions is through laser pulses, because they achieve high peak powers, and is this a lot of energy in a short interval time, and through propagation in an optical fiber, because it allows the light to be concentrated in a very small space, about 5 microns, and over a long distance. However, this type of light can also be generated using continuous light and bulk materials. This is a simplified scheme of one of the sources in pulse regime and based on optical fiber, which is the most commercially used. Basically, the scheme of a supercontinuum source can be divided in 3 parts. The oscillator or CIDR, then an amplification stage, and a third stage where the spectral broadening is generated. The oscillator is the initial part of the system, and Its purpose is to generate light pulses. There are many different schemes for constructing an oscillator. Here you can see a simple design of a fabric period cavity in fiber, where there is a doped fiber between two mirrors that in this case are the sesame, it's a saturable absorber mirror, and a fiber Bragg grating. The design of the cavity will mainly determine the pulse repetition rate, of the pulses, that will be the same that at the end when the supercontinuum is generated. Also, the design of the oscillator determines the spectral bandwidth, the central wavelength. Also, it's important the central wavelength for later generate the supercontinuum, and the pulse width of the pulse duration. The amplifier, as its name suggests, is the stage where the energy of the pulses is increased to achieve levels for generate the nonlinear effects. In the amplifier, as the power increases, nonlinear effects also are generated that contribute to spectral broadening, but it does not have the optimal properties to broadly widen the spectrum. Here, two effects can be clearly observed. One of them is the self-phase modulation, characterized by the appearance of sidebands in the spectrum, and both symmetric, and the Raman effect, which translates into the appearance of a band on the long wavelength side here. Finally, in the third stage, the light from the amplifier is used to pump a photonic crystal fiber. Photonic crystal fibers are fibers which light guidance occurs due to the index difference between the core and the periodic Hall structure surrounding it. The design of the Hall structure, I mean hole diameter, distance between holes, or hole rings with different diameters, etc., allows that the dispersion curve of the light to be designed as a function of the wavelength. This curve has an important role on the nonlinear processes and contributes to optimize the spectral broadening. So this will be a general description of the principles of a supercontinuum laser. Now, to show you an example and see the properties of the light generated, you can see in the next slide the particular case of a supercontinuum developed in Phila. At Phila, we developed one of these SARs a few years ago, and currently we call to this SARs IceBlink. The result we obtain is as follows. We managed to generate light from 450 nanometers up to 2200 nanometers with a very flat spectrum, and with the characteristic peak at 1060 nanometers corresponding to the band wavelength. This is an old graph. This year we have released a new version called Horizon, in which we have made modifications to eliminate the peak at 1060 and have more energy in the visible part of the spectrum. The final characteristics of these SARs are a repetition rate frequency at 80 MHz, a total power greater than 3 watts, and within the visible band, specifically between 450 nanometers and 750 nanometers, the power is greater than 250 milliwatts. The pulse duration depends on the bandwidth considered, since the fiber has dispersion, and its value varies from 10 picoseconds to 200 picoseconds. In the case of 200 picoseconds is when we consider the entire bandwidth. This is a non-polarized source, and the output is presented with a 1 meter fiber section connected to a final collimator that generates a beam with an approximate 2.5 beam diameter in the visible region. Here you have some examples of applications for which our customers have been using this type of source. In fluorescence microscopy, this is the most common application. The advantage is that the wide spectral bandwidth covers the bandwidth of multiple LEDs and single frequency lasers. So with one source, it's possible to illuminate all fluorophores with different absorption bands. And using filters, it's possible to select any specific band from the spectrum. Also in spectroscopy, because halogen lamps are the traditional sources using this field, but the supercontinuum sources are being introduced. Because it offers the same spectral bandwidth and the pulse regime, it has very high intensities and also has a collimated beam output. For biological processes analysis, and fluorescence lifetime is also useful because the 80 MHz pulse repetition rate, it corresponds to a delay between pulses in the order of 10 nanoseconds. And this is adequate for analyze dynamic processes in the picosecond range or few nanosecond range. This repetition rate is not fixed and can be selected and decreased to increase the delay between pulses. Other particular applications are light sheet microscopy, material analysis, photonic devices, characterization, and so on. One thing to keep in mind is that the supercontinuum is a type of source that is spatially coherent, but not temporally coherent, due to the mixture of nonlinear effects that are present. This means that the supercontinuum cannot be compressed to obtain a pulse with a duration similar to the Fourier transform of the spectrum. And this is the reason that this is one of the reasons that make us to develop another model of source that in this case is temporally coherent and allows the pulses to be temporally compressed, close to the Fourier transform. We also call it a supercontinuum source because it has a broad spectrum, although not as broad as a strict supercontinuum. And this is the scheme very similar to the previous one, but with some particular differences. First, the design of the oscillator is different. In this case, it's intended to have a broader spectral pulse. The reason is to later use a delay line, here at the output, to temporarily disperse the pulse. And the broader the oscillator bandwidth is, the longer the temporal duration will be, using the same length of dispersing fiber. In this design, a chipped fiber Bragg grating is used as a mirror to get a broad spectrum. And the output of the cavity is the 1060 nanometers channel of the wavelength division multiplexer. Then in the amplifier stage, by entering with a temporal broader pulse, we can achieve higher pulse energies without generating nonlinear effects that we don't want in this case. For this laser, we want to generate spectral bonding only using the self-phase modulation effect, as the result is a temporally coherent pulse. This graph shows how the total power increases up to 1.5 watts in this case, without significantly broadening the spectrum. Next, this graph corresponds here at the output of the amplifier. And now, as we have temporarily stressed the pulse, we now want to compress it again to increase the peak power and later make the nonlinear effects in the last stage. For this, a fiber with anomalous dispersion, opposite to the rest of the fiber used, this fiber will compress the pulse to about 400 femtoseconds. The fiber is a hollow core fiber. Here you have an image of the structure of the holes inside the fiber. It's special because the core is air, so this allows the peak power of the pulse to increase without generating nonlinear effects because the propagation is in air. At the end of the hollow core, the spectrum is the same that at the beginning, but the pulse width decreases in more than 1 order of magnitude. Finally, in the supercontinuum generation stage, a photonic crystal fiber is also used, but this is designed to have a completely normal dispersion and also flat and close to zero. This ensures that only self-phase modulation appears in the fiber section, obtaining a spectrum like this. It shows how the spectrum broadens symmetrically, like in a self-phase modulation behavior. At the output of the photonic crystal fiber, the pulse duration is approximately 400 femtoseconds, like at the input, but this pulse can indeed be compressed through a compressor to achieve pulses below 20 femtoseconds. The main application for this source is in microscopy. The use of a tunable compressor also allows pre-compensate the pulse for the dispersion that a microscope might generate when the pulse propagates inside, because these are pulses with a large bandwidth and the dispersion of the microscope's affects. So with the precompensation, we can have a very short pulse in the plane of the sample. In the following images, we can see results obtained using this source with a microscope. The first image is a fluorescence image to see a plant tissue of convallaria, where the cell wall and nucleus are visible. Sorry. The second image has been taken using two-photon microscopy, because with the compressor pulse, the pulses are short enough to generate the two-photon absorption, and the image corresponds to a zebrafish. And the third image is not complete because it's a 3D image, and I cannot rotate it, but it has been done with two-photon microscopy and using a confocal microscope. So several images we obtain it at different depths of the sample, generating a 3D image. So this is all what I want to show. If you have any questions.
Thank you, Salvador, for the nice presentation. We now invite questions from the participants. Please use the Q&A feature if you have any question, and we'll forward it to Salvador. And while we await the questions, I have one question, and if I can ask. So you said that for the ice-blink laser, the pulse repetition rate is selectable. Can you briefly explain the technique you use to select? Do you use like a crystal with an RF oscillator or something like that?
Well, we have two models. One is with a fixed repetition rate, it's 80 megahertz. And this is fixed, but we have another model that has a main frequency of 40 megahertz, and you can select multiples from this frequency. You can select 20, 10, 5, because the technique used on the laser is with a pulse picker introduced in the middle of between the oscillator and the amplifier. Because there, if you put a pulse picker, you can select the pulses, and normally a pulse picker is wavelength dependent. So after the oscillator, you only have a wavelength. So it has sense to use the pulse picker there. But if you want to use the pulse picker at the end of the source, where you have a very wide broad band, the pulse picker doesn't work for all the wavelengths, or you can have spatial dispersion. That's why it's better to have, if you want to change the repetition rate, it's better to have a pulse supercontinuum that it has this property, and not do it after the source.
Okay, thank you, Salvador. And we have a question from Salehi, I think. Is the laser you described used for stimulated Raman, I think, spectroscopy? They ask simulated Raman.
Well, I specifically don't know which techniques of spectroscopy is used, because I'm not in contact with the customer normally. But so I don't want to. I'm not sure if I can answer that question. I'm not sure.
Okay, I think we can all thank Salvador for the nice presentation. Our next speaker. Let me move on to our next speaker. Our next speaker is John itself, John Rosas. He's one of the doctoral candidates in the CONcISE doctoral network, and my colleague. And he has a background in electrical engineering. He did his bachelor's and master's in electrical engineering from the University of Costa Rica. And currently he resides in Valencia in Spain, and is pursuing his industrial doctorate at FILA and also in UHI. His research interests are mainly in the manufacturing of ultra short pulse lasers for different applications, such as free space communication and microscopy. And he is also the main organizer of this webinar. So let's I invite John to take over the presentation.
Thank you, Shiva, for this nice presentation. I'm gonna switch from the moderator to speaker. It's gonna be rough, but okay. So you can see my screen? Yes? Okay, perfect. So today I'm going to talk about my PhD thesis that is running at this time. And this presentation, I call it femtosecond laser at 1.5 micrometers for multiphoton microscopy. I'm, as Chiba told you, I'm now in Fila Laser, working with all the manufacturing of lasers, and especially in the R+D department. So first of all, this is my project. My project is inside of this work package, as I told you at the beginning in the introduction of the webinar. And this consists in designing a new microscope. So this microscope consists in obtaining from the sample an image, and have a lot of blocks before obtaining the final result. But my work insides here in the light source. We are working in collaboration with the Universidad Jaume I and the CWI from Netherlands, the CNR from Milan, and us, Fila. So my work consists in designing the laser. For design this laser, we have some different parameters we want in our source. The first of all is the temporal coherence, because this gives us a huge advantage in resolution. The second one is we want a specific spectrum, because we want to excite and select fluorophores. So for this case, we decide to design a source at 1.5 micrometers. And finally, we want a source in the fecond-second regime, because this allows to have a high peak power to make an effective excitation of the fluorescence, but without damage the tissue. So we want a I want to say low average power. But at the end, this allows you to excite the tissue without damage. So another thing we want is that, because I'm doing an industrial PhD, we want to make a product at the end of this project. So we have another requirements, like we want to control the perimeters, we want laser security, we want an easy user communication with the equipment, we want to be confined, compact, and robust, because it's necessary to give our laser to different customers around the world. So everything starts with electronics, because electronics give us the control of the laser, principally in temperature, in the current of the pump diodes to excite the laser, and finally to control all the on-off and the security, because it's going to be a product, so it's have to be saved. With the electronics, we have to decide what technology of laser we want, and we work with fiber lasers, so we decide to start with a laser in fiber. And why fiber laser? Because it's easy to manipulate. We have the convenient light, so it's kind of secure. We have a good quality beam, because the output comes from the monomode fiber. It's very easy to make a compact and robust product with fiber, because all the light is confined and easy to manipulate. And because it's very easy and it's solid, we have a very easy way to cooling, has a long lifespan, and requires a low maintenance. So here you can see a little block diagram about my laser. First, start with an oscillator, as same as Salva told you. And after we have an amplifier stage to obtain power, and then we have a compressor stage to obtain now the shortest pulse that we can. So the oscillator, in this project we also use a factory product configuration, because it's like the house-made configuration in Fila. And we have these parameters, duration pulse of around 750 femtosecond, a pulse repetition rate of 67 megahertz, an output power of 5.5 milliwatts, and the center wavelength is around 1.5 micrometers. This is very nice because at this wavelength, we can use all the commercial components to communication systems, and it's very easy to fuse and to use it in our systems. So at this point, we already have a laser. Oh, very nice. And it's also in femtosecond. Cool. But it has a little problem, and it's we want even shortest pulse duration, and we want more power because 1.5 milliwatts, it's low. So for this, we try to make some different amplifications stages. First of all, a preamplifier, which we going to gain power principally, but also we are going to gain some spectral bandwidth because we needed to achieve a specific Fourier limit transform to get the shortest pulse duration as we want. For this amplifier, we use a kind of amplifier method called parametric amplifiers in fibers, and this allows us to amplify femtosecond pulses with minimal distortion of the pulse. And how is this achieved? Well, using a fiber with a normal dispersion. In this case, this commercial fiber that you can see in the slide, the ER30 4125 from Lieki is perfect for this use. So here we have our spectrum at different power when we increase the current to the power pump diodes. And as you can see, the spectrum is going to be a little bit broader. And the other image on the left, you can see the different shape of the autocorrelation of the pulse. And as you can see, the shape is very close to a Gaussian pulse. So this, in some way, let us to know that the current at temporals is not a distortion. So at this point, with the maximum current, we achieve 100 milliwatts. So we want more. And then we put another stage with a double pump, the fiber, the same fiber as the stage before. And we achieve even more power. With this more power and more broadening of the spectrum, we can obtain now good parameters to compress the pulse because we want the shortest pulse possible to have the highest peak power and perfect for microscopy in multi-photon microscopy. For the compression, we use a hollow core as the same type as Salva chose before. But in this case, this works for 1.5 micrometers and has a very tight bandwidth, around 20 nanometers. So this is going to filter our spectrum that I showed you before. And also with the dispersion of the fiber, we are going to compress the pulse in relation of how many centimeters of the fiber we use, or how many meters. Finally, because it's a hollow core, we don't produce any other nonlinear effects that they can damage or not damage, modify our spectral and temporal characteristics. So this fiber is perfect just to compress the pulse. And finally, we have here the spectrum that we obtain at the end of the hollow core fiber. On the left is around 20 nanometers of bandwidth. And on the right, we have the exactly pulse shape of our output laser. This is a curve that we obtain working in collaboration with the Universidad de Salamanca, because they have a novel method to reconstruct the pulse shape. So as you can see, we achieve 190 femtosecond. It's very, very close to our Fourier limit transform. That is a 130 femtosecond. At this point, we have all the requirements obtained for microscope, and we have a resume here of all the parameters we obtain at the end of the laser, and a few pictures of how it looks, a prototype in our lab bench. Now we just have to prove it, but it's not done yet. We have to go in collaboration with the Universidad Jaume I to try to obtain some images in the few months it comes this year. And that's it for today. So thank you, everyone. If you have any questions, so I'm here. Let me see.
Hi, John.
Hi, Samuel.
Thank you for the talk. Was really interesting. I have a question. Can you reduce more the tempo, the size of the size, no, the temporal duration of the pulse with a compressor?
Okay, so
Or
in this case, we cannot because to compress more the pulse, we need more spectral bandwidth because of the Fourier transform limit theory. So if we want even more shortest wavelength, we have to add another stage at the end to a non-linear stage to generate some kind of supercontinuum at this wavelength. And then we can compress again. And with that, in relation with the spec of the bandwidth we will have, we can compress even more. So this laser is a kind of similarly with the cyclone that Salva showed us before, but a different wavelength.
Okay, thank you very much. Thank
you. Any other question? So we are going to go to the next talk. Thank you so much.
1 second, John. I think Marcos has a question.
Yes, really? But I can see it.
I think he raised his hand. I don't know. You're mute, Marcos.
My mic was disabled.
Okay.
Now, could you explain a little bit more again the compressor and filter that you presented? Like how this study works? How
Here?
the study, the 11 slide.
Yes.
Like how this device works? Is something like this a filter that you?
How this device works?
Yeah.
Okay, so this is a kind of fiber with a huge dispersion. Normally, it's anomalous dispersion, and with the theory of the dispersion, if it's different than the dispersion in the different direction of our pulse, we are going to make the shortest wavelength go even more faster than the long wavelengths. And if you come with a normal dispersion pulse, go through anomalous dispersion, we are going to make a compression. The necessity of be a hollow core is because we are working with a high peak power, and this in another medium is going to generate nonlinear effects not desired for this case. So it's hollow core is inside we have air. So the air has a very low nonlinear coefficient. So the nonlinear effects are not going to appear, and the compression with the dispersion is which are going to make the magic.
Okay. Another question about maybe why can't you work on the 14, 17, 75 nanometer?
Why I work-
in the cross point between the dispersion and attenuation. Is it? I don't know. I'm not like that familiar with this.
Okay,
devices, so yeah, in
okay.
that place, there's an intersect. You would like to be there, right?
No, this intersection is just the red curve is about the dispersion, that you can read the dispersion on the right.
Yes.
So, and the blue are the bandwidth of the hollow core, because the hollow core can't guide all the wavelength, just a few band inside.
Yes.
Because this
Yes.
doesn't work with a normally fiber optic. It's more about another principle of interference. So
Yes.
there are some bands you can work, and for this commercial hollow core has this bandwidth. So they are two different curves of specifications of the fiber, but not a point that I'm working.
Okay. So there could be like another kind of compressors that like standard compressors that they might work at that point, but maybe like the dispersion will be like less used to correct like these
Yes.
non-linearities.
Yes, the dispersion depends on the length of the fiber. So in the right image, I make a study of different lengths of the fiber. And at the point of very close to 50 centimeters is the shortest. It's around 187, very close to the experimental we have.
Okay, but it's mostly flat for that one, and then
Yes, that comes very close. It's because you can see the dispersion is in relation with the wavelength, and also it's very high for few meters. That's I understand.
Yeah. Yes. Oh, thank you.
To you. Okay, so let's continue. Thank you so much. Now I'm going to be a moderator again. And now we have the presentation of Martina Delgado. Martina is a professor from the Universidad of Valencia. She has a PhD in fiber optics, I think especially in relation of resonances, acoustic resonances, and that kind of stuff. She's very good at talking, so I left you the mic. Thank you so much, Martina.
Good afternoon. Thank you very much to the audience who is attending this webinar, and also thank you very much to the organizers for inviting me to participate here. I am Martina Delgado Pinar, as John said. I'm part of the Laboratory of Fiber Optics at the University of Valencia. And today I'm going to talk about one of our research lines, which is devoted to photonic biosensors, in particular, the design of photonic biosensors that use optical resonances for the detection of biomolecules. So I am a physicist. So what am I doing here in the biosensors field? But if you study the parts of a biosensor, you can see that there is a part which is the sensing platform, or where all the binding and interaction between molecules happens. That's the part of the chemist. But there are other parts, such as the transducer and the detector, how we read out the output of our transducer and how we quantify these variations and then convert the changes that are happening in the sensing platform in an output that we can measure, for example, in a voltage. Then that part is where we can do some work. So we are playing a role in the design and fabrication of biosensors. So Here, I'm going to focus on photonic biosensors. I said, of course, right now, the golden standard of photonic biosensors is the surface plasmon resonators, that they also use optical resonances to operate. But I am going to present two other approaches. One, which is based in biodiffractive structures. I will explain later what a biodiffractive structure is. And the other one, well, there are two approaches based on fiber-based biosensors. So when we have to design a biosensor, we have to think about different things. The first one, which is the part that where the chemists have to do their work, is how do we functionalize the surface? That means how we do modify the surface to make this platform, the fiber or whatever we want, sensitive to the interaction or the binding between molecules. And then the other things are how we are going to design and optimize the photonic transducer to make that the sensitivity and detection limit of these biosensors are the best available. And also we have to design the way we are going to read out and quantify the variations that we can measure. So the first example I'm going to show is based on a long period grating, which shows a narrow bandwidth. So you might know that a long period grating is a photonic device that couples the light from the core mode of a fiber to one of the cladding modes. This is resonant, so it happens at a given wavelength, and because the one of the modes implied in the carping is a cladding mode, you can see that there is a fraction of energy around in the outer medium. So then that means that this device will be sensitive to changes in the external medium. Well, so we have our long period grating, and one of the key points of our design is that this long period grating is very narrow in bandwidth, as narrow as 1 nanometer. Normal or conventional long period gratings are usually of tens or hundreds of nanometers bandwidth. So ours is 1 nanometer, which will help us to enhance the detection limit. What we did at first was to functionalize the surface. So this design was done for detecting the hybridization of the DNA. And then we attached one of the strands of DNA. You know that the DNA has 2 strands that they are usually rolled up. And then here we are going to attach first one of the strands. And in this case, what we see is that the resonant peak of the Bragg rating of the long period grating, which was originally at a given point, shifts when we already attach this first strand. So what we are going to do is to read the wavelength shift of this resonant peak, and then quantify the concentration of the second strand that we are going to put in our sample. So what we are going to detect is when both strands roll together, and here you can see in the down right figure that we have a half a nanometer shift when this happens. Then, based on these numbers, we estimated the sensitivity and detection limit of our device for 2 modes, for 2 resonances, and we could see that the sensitivity and the detection limit was improved respect to the work by Chen et al., where they were using broadband long period gratings. This is the first example that I wanted to show you. The second one is based on bio-Bragg ratings. What's a bio-Bragg rating? I'm going to explain that now. The thing is that in this case, we are going to combine one technique that comes from the planar surfaces, which is microprinting of biomolecules. This is already done for chip-compatible biosensors with optical fiber technology. In our case, we are going to use taper fibers. A taper fiber is a fiber that has been reduced in diameter, in our case, down to 3 microns. So over the surface of this taper, we are going to set a microstructure, a pattern of biomolecules. So this pattern of biomolecules will act as a fiber Bragg rating. So to do so, we are going to use a PDMS stamp that we will cover with BSA molecules. We are going to use a model BSA anti-BSA assay, and then the proofs will cover the surface of the grooved stamp of PDMS. When we put in contact the stamp with the fiber, then they will the molecules on the higher part of the groups of the stamp will transfer to the fiber, so we will have this kind of strips of biomolecules. That is what we call a bio-Bragg rating. When we launch light to this device, what will happen is that the evanescent field of the mode that is propagating along the taper will interact with this microstructure of this pattern of biomolecules, and it will produce a reflection that will happen at the Bragg wavelength correspondent to the pitch of this pattern of biomolecules. Later, when we make that these BSA molecules combine or identify the antigen, the anti-BSA, the strips will be higher will be higher, and then the reflection of the light will increase. So here you can see in this FSM image, you can see the actual strips of biomolecules are along a taper of 3 microns. Then, how are the experiments done? First, we are measuring a nude fiber, a nude taper. We put a reference fiber Bragg rating, a conventional one, in a single mode fiber, just to control the conditions of strain and temperature in the laboratory. And then we only see the reflection peak corresponding to this fiber Bragg rating. In a second step, we set the the pattern of BSA molecules, and then we can see at the Bragg wavelength a weak reflection corresponding to this bio-Bragg rating may conformed only by the BSA molecules. So when BSA and anti-BSA link together, then the reflectivity of this grating increases, as you can see, and then we are going to correlate this change in reflectivity with the concentration of the anti-BSA present in the sample that we are studying. So when with this principle of operation, we performed a complete assay, and we could determine the limit of detection and limit of quantification of this device, which are pretty good numbers. Once we had this result, we thought that we could go a step further and then think about another approach to fabricate these bio-Bragg ratings. So we are going to talk now about planar structures because they are chip compatible, and we wanted to make something that was compatible with integrated photonic circuits. And the thing is that when we observe the strips that were produced by means of microstamping, you can see that they are kind of sinuous. You can see meadows along the strips. When that happens, then you have this chirp in the Bragg rating, and this is not the most convenient. So how can we improve that? So instead of using microprinting, we can use our setup to fabricate fiber Bragg ratings with UV light. The thing is that UV light, when you illuminate a molecule with UV light, the structure of this molecule changes, and then you say that you denature these molecules, and they are not active anymore to interact with their antigen or with other molecule that you wanted to detect. So you can, with this setup, you can selectively denaturate the BSA molecules, creating a pattern of active inactive, active, inactive molecules. We tried if we were able to deactivate the molecules, and here you have here in figure A you have the samples, you have the example. The left part was illuminated by affluence of UV light. The right part was not, and then the right part is darker because they are the BSA molecules are binding with the anti-BSA gold label molecules. So once that we knew that we could fabricate these strips of active, inactive molecules, we could expect that we have a better uniformity of the strips that when using microcontact printing, and then we're going to obtain a better beam quality of the diffracted beam. And that's actually what it occurs. We have these beams, the field of the beams of the microprinting setup and the field of the beams of photopatterning. You can see that in the case of photopatterning, they are more well defined, as you can see here also in the profile. With this, we could obtain the limit of the thread detection and limit of quantification of the immunoassay, and we could see that both values were better than the case of microprinting in planar structures and also the microprinting on fiber tapers. Finally, just very shortly, I wanted to show you a very simple device that still it works very well, and then it's the U-shaped fibers. So we are going to take a piece of fiber which is stripped. We take out the polymer coating, and this fiber is a single mode telecom fiber, just the SMF28, the simplest one. So when we bend it, what happens is that the core mode will couple to one of the resonant modes, and it will appear, as in the long period grating case, a resonance at a given wavelength that will be defined by in this case, the curvature of this U-bend. Because it's a cladding mode, again, this will be sensitive to the changes in the external medium. So when we immerse this in water, for example, the resonance will shift in wavelength. And then if something happens, a change in the surface, this will be detected by a means of a shifting wavelength. We have this device that we fabricated to test our biosensors, and then we can see that we have a pie section of fiber and strip, and with a U-shaped and then this is the sensing platform. We perform a BSA anti-BSA immunoassay, and we could see that for a concentration of 50 ppm in 50 ppm on anti-BSA, we could see that there was a shift of several nanometers of several nanometers in wavelength, and it occurs in the first few minutes, 15 minutes, let's say. The limit of detection is not the best, but still, with the simplicity of this setup, is very valuable. So just to conclude, I have presented 4 approaches for different biosensors. All of them are based on resonant phenomena. So compared to the interferometric techniques, it's not ambiguous as the other techniques are, and all of them are label-free performance. So thank you very much for your attention, and I'm ready to answer any question that you may have.
Thank you, Martina, for the nice presentation. We are waiting for the questions in the chat, or you can raise your hand. I have a question in, okay, we have a question from Firoz Cordero. Please go ahead.
Hi, yeah, very cool. I was wondering how difficult or limited are you by the molecules that you're able to use to functionalize like the fiber?
That's the part of the chemists. But actually, it's silica. The fiber surface is silica, so it's not there are quite well-established methods to functionalize different molecules. I cannot tell you if there is something that cannot be functionalized to silica, because I'm not an expert on that. But I know that on silica there are these how they call it, click and I don't remember the name, but different techniques that can functionalize a large variety of molecules with not simple for me, but simple for them techniques.
Okay, so just as a follow-up, so as of now, like of the different processes that you may have been interested on sensing, you've never have faced not finding a molecule that can functionalize silica, or have you have that been a?
You mean that if not attached to the surface?
No, I mean like if, let's say, I don't know if you've thought of sensing different processes, and I guess for different processes or molecules, you would need, in some occasions, different functionalizations. Yes,
sure. I mean, if, for example, we have used this UV denaturing and this the tapers as well, used for example, protein C or other things. And then what happens is that the specific functionalization that you need to do at the beginning is different depending on what you want to detect. Because, for example, for DNA, you put a strand and then detect the other strand. For BSA, you put BSA and then detect anti-BSA. But if you want to measure the vitamin C, then you have to put the specific anti-protein thing for that protein. But I'm not an expert on that kind of things, I'm afraid.
Yeah, thank you.
Thank you, Victoria. We have a question, I think, from Enrique. Please go ahead.
Very nice presentation, Martina. Thank you. I have a question about the last method. I mean, do you bend the fiber? I wonder if you optimize the bending curvature to the application, and you
yes. Where if I can let me show you. For example, here, can you see this one?
Yes.
So here you can see that we measure this for different radius.
Oh, okay.
For different radii, and then also with different external index, we also try to see how they behave and this kind of thing. So we tried different lengths of the strip section as well, but that was not so it was not so important. So the best the important thing was this to select properly the radius because we wanted to have the resonances at a given wavelength. Mm-hmm. So, and also the cool thing is that the linear performance worked the same in air that in water. So there was not going to be a different behavior when we were using water or air or these kind of things.
Thank you.
Thanks everyone for the questions. Let's thank again Martina for the nice presentation. Thank you for joining this webinar. Let's move on to our next speaker, who is Enrique Tajahuerse. He's a professor of Optica in Department of the Física, and the rest is in Spanish, I'm sorry, from UJI. And he has a background. He's a doctor in physics from the University of Valencia, and he's a coordinator of optical research group of UJI, and he's also a fellow of Optica. His research interests are mainly in diffractive optics and adaptive optics, security and encryption, digital holography, and currently he's working on computational imaging with structured light and compressive sensing. Thanks a lot, Enrique, for joining the webinar, and please go ahead with the presentation.
Okay, thank you very much for the introduction. And as you mentioned, my name is Enrique Tajahuerse, but the work I will describe today is also supervised by Professor Jesus Lancis. We are both at the Photonics Research Group and the Institute of New Imaging Technologies at University Jaume I. Today I will talk about computational microscopy with single pixel detection. Of course, there are many other researchers involved in this work. Two former students that finished the PhD here some time ago, Fernando Soldevila and Armin Lenz, and the current students, Luis Ordóñez, who will finish the PhD this year, and Everley, Tobon, and Samuel Zapata that are involved also in the CONcISE project, as you know. And also, of course, we have very strong collaborators. In fact, many of the experiments I will explain today were performed in the laboratory, which are Alberto Greci, Andrea Farina, and Cosimo D'Andrea from Politecnico di Milano. Of course, you know that Andrea, in fact, is coordinating the CONcISE project. Okay, so I will describe some computational imaging techniques and apply them in microscopy. I will show how easy it is to perform dual microscopy and four-dimensional microscopy, how to increase the capabilities of a microscope working with single pixel detection with optical sectioning and super resolution, and finally, our last achievement in autofocusing. Let me tell you a little bit about the single pixel camera. You know that in conventional imaging, we can take an image with a camera of an object in a single shot, because each pixel of the camera corresponds to a small spot in the object. Mathematically, this can be described with a measurement matrix, which is a diagonal, because you know each point in the vector object corresponds to one point in the vector image. But we don't need to use cameras always. We can also use a single pixel detector or a bucket detector. For example, by scanning the object with a raster approach. No? This is very common in confocal microscopy or other approaches. Now we transform the object into a temporal signal, and from this signal we can reconstruct the object. It's interesting to recognize that mathematically the measurement matrix is the same. It's again a diagonal, because each point of the object corresponds to one point of the image. And this makes very important not losing any measurement. We need to scan all the object. But this is not what people usually call single pixel imaging. In single pixel imaging, the object is illuminated with wide field, with a structured wide field illumination, as I am showing here. And a single pixel of the bucket detector collects light from all the area, which sometimes is more convenient. Again, we perform a sequential acquisition of information, and mathematically, the measurement matrix is very different, because now with each measurement, each row of the matrix scans the whole object, which sometimes is very, very convenient. If we use Hadamard patterns to scan the object, which is a special orthogonal transformation, in fact, we are doing a Hadamard transform of our object, so the image of the object can be easily recovered by performing an inverse Hadamard transform of our measurement. Okay, so why to do this with on imaging? Why to use single pixel detectors? There are several advantages. Sometimes efficiency is larger. Also, the simplicity of the sensor, it's only one pixel, allows to measure in ranges out of the visible one, for example, in terahertz, is very important. And also, because of the simplicity of the sensor, it's also very easy to measure different parameters of the light, such as the polarization state or the spectral content or to measure time of flight. The main problem of the technique is that we need time because we have a sequential acquisition. But this can be compensated with compressive sensing approaches or other methods. We have been working in many applications along the last years, such as holography, imaging through scattering, or diffuse optical imaging. Today, I would like to focus on microscopy applications because this is also our main subject in the CONcISE project. Almost all our setups are based in the same structure, as you can see here. We illuminate with a parallel light beam our digital micromirror device, which codifies Hadamard patterns, and those patterns there are projected onto the field diaphragm of our microscope, and then with the tube lens and the microscope objective, they are projected onto the sample, as I show you in this simulation. So the sample is scanned with these wide field patterns in a microscopic scale. Then we can record the transmission light, the reflection light. We can record the fluorescence coming from the sample. If we record the reflective light, the approach will be this. The reflected light will be collected in our bucket detector. The signal will be a temporal one that, with the proper algorithms, is transformed into our image, and the image of our object. Let me show you how simple it is to perform dual microscopy with a commercial microscope. The only thing we need to do is to illuminate our digital micromirror device, our spatial light modulator, and with a high-quality optical system, project the patterns onto the field diaphragm, which is here. Now the microscope works without alignment, without any kind of alignment, perfectly focusing the light patterns onto the object. If we collect the light transmitted by the object, we will recover a transmission image of our object. If we collect the light reflected, we will reconstruct an image of the object from the reflection light. And we can do both at the same time. And this is interesting because there is a perfect matching between any point in the transmission and the reflection image, something that is very difficult to do with two cameras in a conventional microscope. Those are a couple of examples of images taken by reflection and transmission of two insects that we can compare with the image taken with a conventional. Let me show you that it is possible to take multi-dimensional images with microscopy. This is a collaboration with a group in Politecnico di Milano. The idea is to get an image in 4D. We will have two spatial coordinates, x, y, but also spectra and time. And we will do this by data fusion, by fusing the image taken by a conventional camera with a high spatial resolution by a single pixel low resolution multispectral camera, which is able to record a collection of low spatial resolution multispectral images, and also a low spatial resolution single pixel temporal image of the object. Let me show you how the system works. First, we illuminate a fluorescent object with a picosecond pulses from a laser. The object has 3 fluorophores, each one with a different spatial distribution, a different spectral distribution, different time of life in the fluorescence. And then we record the 3 images. Conventional image, monochrome image with a high resolution camera. Then second, single pixel image, a single pixel imaging system will allow us to record 16 spectral channels with low spatial resolution by using the digital micromirror device. For each Hadamard pattern we project into the spatial light modulator, we record 16 spectral channels, so we are able to reconstruct these multispectral images. And finally, in the second output of the digital micromirror device, we record for each Hadamard pattern the time evolution of the fluorescence. So we are able to record like a video, different temporal frames of the low resolution images of the fluorescence. So with these two low resolution spectral and time images, and with the high resolution in space, we combine all this information. I will avoid the mathematics of the data fusion algorithms, and we recover an image with high spatial resolution, the wavelength and time of our object. Here you can see the result. I am showing here the 16 spectral channels in time, how they evolve in time. And here on the right, you can see the time evolution of the different letters, different fluorophores. After this proof of concept experiment, also in the Politecnico di Milano lab, under the supervision of Andrea Farina and Cosimo D'Andrea, we did the microscope experiment. So we did almost with a similar setup. We got to obtain high resolution multispectral images of cells by combining high resolution conventional images taken with cameras with low resolution multispectral and time images taken with single pixel cameras. You can see how the resolution is improved from the low resolution images to this one. We can see here a magnification of an area, and we can select any point on the image and see how the wavelength is, the spectral distribution, and the temporal behavior of the fluorophore. Okay, now let me show you how we can add in single pixel microscopy optical sectioning and super resolution. This is very easy because we use a very well-known technique called structure illumination microscopy. Maybe you know that when we project intensity masks, if we project gratings onto an image, then because the gratings are very sensitive to defocus, we can see the gratings only in the areas which are in focus, but we don't see the gratings in the areas out of focus. So by using several masks and applying a very simple algorithm, by taking several images with phase shifting, we are able to recover images that contain only the well-focused areas. We call this optical sectioning. You see, we remove the out-of-focus information. So we did this in microscopy. To this end, we project, again, Hadamard light patterns through the field diaphragm, and we reduce it, project it onto the samples through the microscope, and we record the bright field reflected light or the fluorescence coming from the sample into the photodiode. This is our single pixel microscope. It's very interesting that you can see we have areas in focus and areas out of focus when we put here a 3D sample. By using the structure elimination microscopy technique, by adding gratings to these images and using the SIM algorithms, we are able to get only sections, areas of the images which are located at a fixed axial distance from the microscope. And with a collection of axial images, we are able to recover 3D information from the object. Please pay attention that, and this is very interesting, to project gratings onto the object, we use gratings close to the photodiode and not in the spatial light modulator. This is, I have no time to explain this, but it's very, very, very interesting. This is what we do. We project Walsh Hadamard patterns onto the objects. We combine them with the gratings, apply phase shifting algorithms, take a collection of images with the gratings with different phases, and finally with this we obtain the optical sectioning. all this done with a single pixel detection. Here are other examples, how with optical sectioning we are able to record only some information at a fixed distance from the microscope, and also that the system works in fluorescence also. Okay, SIM techniques are also used for super resolution, but the grating we are using here to obtain optical sectioning is not useful in this case for super resolution. And take into account that we lose resolution in a microscope, in a single pixel microscope, because the Hadamard patterns we project onto the sample have high frequencies that don't go through the OTF of the optical system. And then, so the patterns are projected defocused, and we don't obtain the image properly. So we need to compensate for this. We need to compensate for the limit in the frequency projected or by the microscope onto the sample. And we can do this by adding a second grating here in front of the digital micromirror device. So this grating will generate high frequencies from our Hadamard patterns that will go through the microscope objective. By doing this, by adding here, together with our Hadamard patterns, gratings, and also gratings at the output, if we want optical sectioning, we have proved that it is possible to get super resolution to improve the resolution of single pixel microscopes. If we compare our technique with the conventional one, you can see the improvement. This improvement can be seen also in those images on the right that improve the image, the conventional single pixel imaging. We can add also optical sectioning. And we have proved that the improvement in resolution can be applied also to fluorescence. And I will end adding some new features to our microscope. We have working recently in autofocusing techniques, because it is very important to focus the Hadamard light patterns onto the sample. If the Hadamard patterns are not projected properly, it doesn't matter if the sample is well projected. We will obtain defocused images. So to improve this and to focus automatically, we have designed a method. We have designed a method to use only a few coefficients in the Hadamard space to define a merit function, and by using this merit function, we analyze if the image is well or not well defocused. And to change the focus, we add a tunable lens onto the aperture diaphragm of our microscope, so we can change automatically the focus position of the light patterns. With this, we have proved that after just a few iterations, it is possible to get the microscope to achieve the perfect focusing. I will finish saying that we have built in our lab a new platform for single pixel microscope that has been finished recently. In fact, we are going to publish like a tutorial explaining how to align the illumination port, the projection setup, the transmission and the reflection setup of the microscope that will be published in scientific reports. Our paper has been accepted just this week. And with this microscope, we have proved we are able to get also reflection and transmission images of different materials and biological samples, such as epithelial buccal cells and red blood cells, both in visible and infrared light. I will skip conclusions because I think this was too long. Let me finish saying that with this platform that we have now in our lab, we will now collaborate with John Roses. We will attach the laser that is being developed in FILA. Also, we are collaborating with CNR in the adaptive optics system that will be attached to the microscope, and with CWI in the algorithms and the AI approaches that we will use to configure the whole microscope for performing nonlinear microscope. So this is all. I will finish with the acknowledgement to the group and the funding agencies. Thank you very much for your attention.
Thank you, Enrique. Amazing talk. A lot of knowledge in microscopy. So now if we have questions, this is the moment. Anyone? Here, I think we have a question. From Twardowski, asking, what are the constraints on the DMD resolution?
Well, we have two constraints, one geometrical and one given by diffraction. The geometrical one is given by the size of the pixels of the micromirror device, which is around 13 microns in size. We can, through the microscope, by using a 40 per microscope, we can reduce these pixels a lot, let's say to 300 nanometers, something like that. But there is also the limit given by diffraction. You know, light is diffracted in the apertures of the microscope. So the final resolution is limited by the aperture of the microscope. And for the conventional microscope, this can give a resolution of around, let's say, 800 nanometers. So no matter how small you make the pixels of the micromirror device, diffraction will not allow you to get high resolution images. So you have both restrictions, geometrical and given by diffraction.
Thank you, Enrique, so much. So because we are delayed, we are going to continue with Victoria. Thank you so much, Enrique, again. And now, well, Victoria is a doctoral from, let me see, just to remember. Okay, so she has a degree in physics and electrical engineer from the University of Costa Rica. She has a PhD in material science and engineer from Georgia Institute of Technology. Now she is working as postdoctoral research scientist in the Columbia University, and the research interests are emerging material from optics and photonics, nonlinear optics, and ultrafast spectroscopy. And also she was my colleague in the university. So thank you so much, Victoria, to be here, and I'll let you the mic.
Hi, John, and everyone. Thank you for the introduction and the opportunity to talk. Since we're a little late, I'm going to just dive into it. The title of my talk is Tracking Multiparticle Interactions in Time and Space. It might be a little generic, and you'll see that it falls within a little bit of a different realm that's not as instrumentation centered or biologically centered, but we're going to go slowly into it together. Okay. So first, I want to, from a very broad audience perspective, clarify what we can think of as multiparticle interactions. So you can think of, at the most basic, multiparticle interactions as collisions, as in classical mechanics or these billiard balls. In chemistry, when you have a proton and an electron that come together to form a hydrogen atom. And also, for example, in solid state, when you have metals and you excite them with a laser or any light, and then you create this sea of oscillating electrons. So my research actually falls within the realm of solid state, not in metals, but specifically in semiconductors. So what we do is that we want to track particles and quasi-particles in semiconductors that are created after light excitation and have very short lifetimes within the range of femtoseconds to milliseconds. So this is how like the energy landscape of a semiconductor looks like before you excite it with a pulse of light and you promote one electron from the valence band to the conduction band. And when you do that, you leave a hole behind, and a positively charged hole. So these are the two particles that we will have in semiconductors, electrons and holes. And now I want to talk a little bit about the term quasi-particles. So one of the most basic quasi-particles that we find in semiconductors are excitons, or electron and hole pairs bound by Coulombic forces. So sometimes when you promote that electron, despite they're at different energetic levels, they're still physically close enough so that they can still be attracted by Coulomb. And that forces the electron and the hole to move together around the material. So in some situations, or in a lot actually, we can treat it as a unique particle. But since it has different components, we call them quasi-particles. And this can become even more and more elaborate in solid-state semiconductors, where sometimes you can create a charge carrier that distorts the lattice like this. So in this case, this electron attracts these positively charged charges around and repels these. And that electron, when it moves around, it moves that lattice distortion with it. So this, it's called a polaron in this case scenario. And we have the same the counterpart quasi-particle, where an electron and hole pair, or exciton, strongly couples with a photon that is confined in microcavity or metasurface. So we can discuss the details of how these quasi-particles form in the questions, but the main idea I want you to take from this is that it's a bunch of things that move together. In this case, it's a charge carrier with a distorted lattice, and in this case is an exciton and photon that actually couple so strongly that you change the energetic levels in the system. So you create these upper and lower polariton bands that are different from this exciton energy and this microcavity optical photon energy. Okay, so before diving into the techniques and our observables, why do we care? We care because these quasi-particles, particles, and their interactions have profound implications in electron mobility, energy harvesting. We can use them for light-based computing and also for sensing. And I want to just briefly mention the point of sensing, because nowadays we have semiconductors, especially 2D semiconductors like graphene and transition metal dichalcogenides, that can be as thin as 1 atom thick, and you can use them to sense the dielectric environment of a sample that you put on top. So I just wanted to make that point because I think it might be of interest for the audience. Okay, so these multi-particle interactions, like these collisions or attractions or repulsions, most often manifest as energy shifts. So if you excite, this is an example of excitons. So your exciton at low laser fluences has this energy of around 2 electron volts. But as you increase your laser fluence, you can see this red and blue shift. So you can associate that blue and red shift in a lot of cases to curves like this, where you have repulsion and attraction if all comes from Coulomb. I'm making this point because the first way to track multi-particle interactions in time is to make use of this observable and track then this energy shift in time before we put in our spatial variable. So one of the most common techniques is pump probe, in which you come with ultra-fast pump, you excite your sample, create all these quasi-particles, and then you come with a probe that will just be there to sense these energy shifts around. The key ingredients, as you can see from these type of setups, are that we need an excitation that is shorter than the particle lifetime, or shorter than the process that you want to measure. And we also need a broadband probe that will cover the spectral range of the multi-particles and their energy shifts. Okay, so we control this time delay too, so that we see the evolution of these energy shifts in time. And what we get are plots like this, where you have your energy here, your time delay here, and you can see in here how the energies of your system shift in time and go from the energies of an exciton to the formation of the polaron. And this was the case of like coupling with the lattice that I was showing you earlier. We can also think of more elaborate systems. So in here, this is a system where we have exciton polaritons, which are those mixed exciton light states. Just from now, the only thing I want you to take from this is that we have these multiple states marked in white lines. And when we use more elaborate techniques like this 2D spectroscopy, what we have is that we can resolve the energy at which you excite the sample. In this case, it absorbs 2 photons, so it's double the energy, and the energy of the emission. And then you can see in the diagonal when you have states that are associated to 2 particles. So you can see here that this state in the diagonal is associated to 2 lower polaritons interacting. So we have it right here. And then we also see these 2 peaks in this cut here. So this cut, it's this cut, and we see these 2 peaks, which correspond to also interactions between the lower polariton and the excitons A, and in this case, the lower polariton and middle polariton. So this is, I guess, there's a lot of detail here that we can happily discuss, but the main takeaway is we can obtain a lot of energy of multiparticle, a lot of information of multiparticle interactions from energy shifts. And nevertheless, well, never the energy shifts resolved in time specifically, but nevertheless, there are some limitations. And the main limitations are that we cannot really correlate the morphology of the semiconductor with the processes observed. And also, we can only track particles and quasi-particles that have very strong manifestations in reflection, transmission, or photoluminescence. So I'm gonna show you a technique that we use here at Columbia a lot in my postdoc. Just as a context, most of these multi-particles interactions resolving time was more of the work that I did during my PhD. And now I'm moving to also add this space ingredient into it. So we use this technique called a stroboscat, in which we have a confocal pump and a wide field probe. And the whole idea is that we collect both the reflected and scattered beams. And the reason why we have a reflected beam is because we use a cover slip on top of our sample. So you have a first reflection between your cover slip and your sample, and then a scattering within your sample. And those 2 beams, or yeah, let's call them beams, will interfere within them, and that's what we will detect. And then on top of that, we turn off and on this confocal pump like we did before in pump probe, so that we can have a time resolution. So the difference in time between this confocal pump and that wide field probe will give us information in time of how these quasi-particles propagate. So nicely, this technique is non-invasive. It's based on non-resonant interferometric scattering and allows us to track any changes in material polarizability and refractive index. So what does this technique images? It images evolving particle distributions. And you can think of evolving particle distributions as what happens when you put a droplet of ink in water. You create your particles, and then they will propagate in your material. And in some cases, they'll found boundaries, membranes, or whatever, and that's kind of what we image. So now I want to slowly show you a broad range of materials and different quasi-particles that we've measured using this technique. The first have been electrons in halide perovskites. What happens is that we create a population of electrons, and then we see them expand in time as that droplet of ink, and that's kind of what we track. But I've mentioned you in the introduction that in these materials or these semiconductors that we study, there are multiple different quasi-particles that we're interested on. So here I'm going to show you some data on other quasi-particles. So in this case, this is a transition metal decalcogenide. What we do here is we also create some excitons, but we see a very different thing going on. We see these coherent rings here, like expanding out. So actually, after some analysis, what those represent are that you are creating your excitons, and then that creation of excitons is creating these oscillations in your lattice. And that's what we're imaging right there. We can also image funneling of excitons into potential traps. So in here, we created also a pulse of excitons in this same material with some bubble defects. You can see the defects here in this AFM. And then you see that those excitons propagate in time and funnel specifically into the positions of those nanobubble defects. And those nanobubble defects have this effect because of potential confinement, but we can also discuss that further. Here, one of our latest works was in polarons. So again, we created some excitons that when they coupled with polarons, the contrast became bright, as you can see this evolution here from the center to the side. And then we can measure how the polaron propagates. And one thing that I want to highlight is, since all of these cases that I've shown you as of now are very exciton-based or electron and hole pair based, you can see that all these timescales are within the nanoseconds and so on. But in the case of exciton polaritons, we also have, or exciton polaritons are the shortest lived quasi-particles that we work with. They're often within 1 picosecond lifetime. So we need shorter pulses to be able to image this propagation and operate at a significantly shorter time regime. But nonetheless, we managed to do so, and we can also image their propagation here. So the reason why they're faster is because you can think of these as part light, part matter, and their part light makes them really, really fast. As you can think, I guess, intuitively think from the group velocity of light in the different media. Okay, so I'm going to show, I've shown you a bit how we track them in space and time and all these different quasi-particles and particles, but I want to now discuss a bit how we obtain information from multi-particle interactions like collisions and such from these evolving particle distributions in time. So in this example, what we do is that we can track the mean square displacement as a function of time. So you can fit Gaussians to the different populations. So in this case, the dark spot is one of your populations. The bright spot is another of your populations because they correspond to different particles. And you can, if there were no interactions at all, you would expect a normal diffusion. But you often have interactions, actually. They're very ubiquitous. So they lead often to subdiffusion behavior with time, like this line here, or to ballistic or superdiffusion propagation. So actually, as you can see here, if we track just the excitons, they have this MSD response, but when we trap the polarons in this material, we have superdiffusion. It's not ballistic because it's not the power law with time is not a square power law, but it's quite fast, right? It's around 1.7, and here you can see the distributions of exponents that we've measured. So this gives us a lot of information of the interactions between these quasi-particles. In the blue curve, we have information about interactions between excitons in WS2, SE2, and in the red curve, we have interactions between these polarons in this material, which are all of these that I'm showing you here. And we can see how having that exciton coupled with your lattice and forming a polaron enables them to travel quite fast. Meaning, or we can think of them as exciton, like kind of surfing on top of your lattice oscillations. And I want to also emphasize a bit the case of polaritons that I've mentioned. So I mentioned that when you have this light matter hybridization, you have these additional states here besides your excitons. So we can probe both your excitons and your exciton polaritons that coexist. In this case, we specifically probe the exciton polariton here. And we see how the excitons show pure exciton transport like we expect in the nanosecond regime, and the polaritons show very fast transport in the femtosecond regime. And when we track the mean square displacements of all of these particle distributions, we see subdiffusion for the excitons and superdiffusion for the polaritons. And we can even calculate the velocity at which they propagate. So I guess there was a lot of technical detail that I left a little bit out, but I wanted to show you how we image these different wide variety of different quasi-particles in semiconductors in solid state. How we need very fast, ultra-fast pumps that are shorter than the particles and processes that we want to probe to be able to see them. And I hope I can convince you that being able to image these propagations in a very simple way and very like natural way, as you can also see just ink propagating in water, gives us a lot of information on the multi-particle interactions from a very intuitive point of view. And with that, I just want to acknowledge all the people that I'm working with now, the Lore Lab, which I guess they're mainly the ones that have implemented this technique, Straubos Cat. Then the Shock Lab, which is my other lab, we do a lot of sample making and also mainly nonlinear optic processes. And I'm happy to take any questions.
Okay, thank you so much, Victoria. It was an amazing talk. Okay, we are now open for questions. For meanwhile, if someone have a question, I have one. It's more about the pump source you use. That's because I'm interested in lasers. So for this image you get and photograph about femtosecond time, what kind of source do you use?
Yeah, so we have a carbide and an optical parametric amplifier, and we do further pulse shaping. So this is a 100 kilohertz repetition rate, and we do further pulse shaping outside. So what we typically do is that we measured our, I guess we do frog, and we use either, there are different options, but it can be prisms, it can be chirp mirrors, or it can be a spatial light modulator to further compress the pulse after the output. So we do this in free space. So like we have our laser output, we compress it as short as we can. And that's what we use, yeah. For honestly, like the work I've showed here has like a broad variety of sources because for the case of 2D spectroscopy, which was without the spatial resolution yet and more focused on the energy, in this case, we didn't really care that much of having a broadband. So actually we use, like we broadened the pulse with a nonlinear optical parametric amplifier, and then we compressed it. Yeah.
Nice. Okay, yes, a lot of sources at the end.
Yeah.
And now we have a question maybe from Luis Jorge Poveda.
Yes,
you
hello.
can speak now.
Can you hear me?
Yeah.
Yes.
I'm sorry because I don't have the chat active. I don't know why. But okay, I will just ask the question. It's about the simulation techniques you use. Oh, first of all, thank you so much for the talk. It was really interesting. But I wanted to ask about which simulation techniques you use. I don't know if you use finite element analysis, or do you use some sort of mixture of different simulation techniques for studying these kind of interactions?
Yeah, that's a great question actually. And maybe I would say sometimes there's a lot of theory work, but then finding theory work on the actual experimental conditions that we operate on, it's a challenge. So for 2D spectroscopy, for this case that is not a microscopy base, there's a whole line of development through perturbation theory in quantum mechanics and Feynman diagrams that you can use to track and predict these energy shifts.
Mm-hmm.
In the case of the spatial, here it gets a little bit more tricky. We have to do a lot of Monte Carlo simulations using kinetic diffusion equations because when you go to more complex diffusion equations, you stop having analytical solutions. So that's what we've done as of now, but I would say there's definitely room to play around with how we simulate, especially, I guess, as you've seen, we're very careful, and spatial distributions in time that we report, we make sure that we can model them, right? But I'm not gonna lie, and we found materials that we haven't yet been able to model, and yeah. Especially depending on like how complex your material gets. So in this case, these potential traps was very nice. We were able to model it, but like as long as you start adding like more interfaces or grain boundaries, or then things can
get messy. Yeah. Thank you.
No problem.
Okay, thank you,
Just
Luis.
as a comment, maybe if you're interested, this paper, the one on the polarons, have some info on our Monte Carlo simulations for these diffusion.
Oh, okay. I'll take note then. Thank you.
Okay, so now the last question, Marcos, do you want to proceed?
Sorry. Thanks for the talk. Or the previous slide where you were showing like this kind of potential traps. This is another question because it's in the funneling. So these traps they form randomly over the sample in some way. You don't topologically like dispose them.
Yeah, that's a great question. So in this case, since we're working with these transition metal dechalcogenides that are very, very thin.
Yeah.
So they're very, very thin, and sometimes when you just move that very thin crystal, you can stop thinking of it as a crystal, and it's more like a paper sheet. So when you deposit it into a substrate, you form these nanobubbles. So in this case, we were just using those nanobubbles that naturally form sometimes when you move these crystals from one substrate to another, or when you exfoliate like an atomic thin layer into a substrate. But you're absolutely right that I mean, this was great for knowledge, but ideally you would like to control these potential traps that you form. We've tried different like trying to create these nano bubbles intentionally with deposition in like specific humid conditions, but other things that people in the group work, it's not my work, is this author's work, Hao and Su. She's been working with creating quantum dot arrays as controlled potentials for controlling funneling of excitons and other stuff.
And that's your, sorry, I will make another question, but sorry if it's fine, then you just skip me. But then your group does like theory and experiments. So if you, I guess that this model, so like if you're using disorder or some sort of that will be like way more easier than the other methods that you were describing, like using experiment, the diagrams, all that, to describe this kind of shifts on the subdiffusion and the subdiffusion. So I don't know if your group does mostly like experimental work or
we are mostly, I would say we're mostly experimental. Yeah.
Okay.
Yeah, I would say we're mostly experimental. A lot of these, like all these techniques, home built. I notice a lot of you do a lot of fiber-based work. All of these are like mostly in air, except maybe some small parts where we like guide the collection of light of fiber to the detector. But like most of these are like free air setups. And yeah, we do mostly experimental work. I would say we try to collaborate with some people. There's some simulations that we do on ourselves, especially if our theoretical collaborators are a little slow. But yeah.
No, from the experimental results that you have is like getting to know this kind of shifts on the MSD is quite involved, but then they analyzing or trying to impose models on top of that. Yeah, they have to be like check or one trace. But thank you.
No problem.
Okay, so we are done. We did it. Thank you so much to everyone to be here in the first webinar of CONcISE. Thank you so much. See you in the next webinar in a few months. Arigatou gozaimasu.
Bye. Thanks for the opportunity.
Bye,
again.
thank you.
Thank you.
Bye.
Everyone, bye.
Bye.
Bye. Have a nice day. Thank you.